Welcome back to another episode of Gene Expression. In order to get information from DNA to protein, all three types of RNA must be used. In addition to this, the two processes of transcription and translation must occur. Today we'll be discussing how RNA molecules are created from DNA by a process called transcription. So what is RNA? RNA is ribonucleic acid and its monomer is nucleotides. A nucleotide consists of a nitrogenous base, ribose sugar, and a phosphate group. RNA can exist in three forms: messenger RNA or mRNA, ribosomal RNA or rRNA, and transfer RNA or tRNA. Like mentioned, all three forms must be used to code DNA into proteins. RNA is very similar to DNA, but there are three significant differences between the two. First off, RNA has ribose sugar, while DNA has deoxyribose. Secondly, RNA is single-stranded, while DNA is double-stranded. And finally, RNA has the nitrogenous base, uracil, instead of thymine. RNA molecules are created by using DNA as a template. Transcription requires the use of RNA polymerases, which read the DNA template in the 3 to 5 direction. RNA polymerases are special in the sense that they can start the transcription process without a primer, unlike DNA polymerases. RNA polymerase will first start off by attaching itself to nucleotides and unzip the DNA helix. From here, the polymerase will add the RNA nucleotides complementary to the DNA template strand, pairing adenine to uracil and cytosine to guanine. There are three processes in transcription, the first being initiation. In initiation, the RNA polymerase will bind to the promoter. A common type of promoter in eukaryotic cells is a Tau box, which would be slightly above the starting point. A starting point is a nucleotide area where the RNA synthesis actually begins. Transcription factors will be used in eukaryotic cells since they help mediate the binding of RNA polymerase and initiate transcription. The whole complex of transcription factors in RNA polymerase II bound to the promoter is called the transcription initiation complex. Next, the DNA strands will unwind and the polymerase will initiate RNA synthesis at the starting point on the template strand. The stretch of DNA downstream from the promoter that is transcribed into an RNA molecule is commonly known as a transcription unit. Step 2 is elongation. The RNA polymerase will move downstream, unwinding the DNA and elongating the RNA transcript in a 5 to 3 direction. The new RNA molecule created will gradually peel away from its DNA template, and the DNA strands will reform a double helix. The final step in transcription is termination. The RNA polymerase will reach a stop sequence of nucleotides. This sequence is AAU-AAA. The RNA transcript is released, and the polymerase will detach from the DNA. The DNA will form itself into a double helix again, and the single-stranded RNA transcript will be formed. The pre-mRNA will need to go through one more step before the genetic message is dispatched into the cytoplasm. RNA processing will take place, which involves both ends of the primary transcript to be altered. The 5' end will receive a 5 cap in the form of a modified guanine. The 3' end will be altered with a poly-A tail. Both ends have similar functions in the sense that they protect mRNA from degradation and help with the export of mature mRNA. In order for the pre-mRNA to turn into mRNA, spliceosomes and riboenzymes will carry out a process called RNA splicing. In this process, spliceosomes will catalyze the cutting of the pre-mRNA and the splicing together of the exons. Exons are the sequences that are expressed. After these processes, mRNA will be ready for translation. Thanks for watching. Have a magical day.